Hi friends, hello everyone. Welcome back to Thanmaya tutorials. You go to our channel for a clear and simple NCERT diagram explanation. In this today's video, we are diving into a chapter called as Sexual Reproduction in Flowering Plants from Class 12th Biology. We will be breaking down each diagrams step by step so you can easily understand the concepts, as well as I will also see which are the diagrams that you have to practice and learn for your exam point of view. So grab your textbook and keep aside and get started. The first diagram which is there in your textbook is the diagram of flower, that is longitudinal section LS of flower. So in this flower you should mainly understand the two things. One is the accessory organ, another one is a reproductive organ. So in the particular flower we have two organs. Accessory organ includes your calyx and corolla, that is your sepal and petal, which is used for only the protection as well as the attraction for the insects for the case of pollination. And next part is reproductive organs, which includes male reproductive organ, that is androecium, and female reproductive organ, that is gynoecium. So androecium consisting of two parts, one is anther, another one is filament. See, this is an anther and this is a filament. Okay, whereas female reproductive part is called as pistil, that is gynoecium, which consisting of stigma, style and ovary. So this is a very easy diagram. You can easily learn this diagram. I think hope you understand all the labeling also and the drawing also. Now let us go to the second diagram. The second diagram is a typical stamen. So, and also they have shown the three-dimensional cut section of anther. This is they will not ask actually for drawing and all, but simply you have to see for your competitive exam wise. You can see I have already said anther will be there, filament will be there. Filament is a stalk-like structure which is helping in the to hold the anther. Okay, and we know that anther. A typical angiosperm anther is a bilobed, and each lobe will consisting of two thecus. See, can you see the bilobed? This is one lobe and this is one lobe, bilobed. And can you see in each lobe we have two thecus? That is why it is called as dithecus. So hence it is a bilobed and they are dithecus. Very important. And we have a line of dehiscence, that is where the pollen grains, when it is matured, it will rupture. Okay, that line will get a rupture and pollen grains will come out. Okay, I think hope you understand about this diagram. Now let us move on to the next third diagram. So the third diagram, which is very important diagram. So this you have to practice. Actually in our examination, that is 2025 examination also, we have got the same question where we have to draw this diagram and we have to expand this. See, whenever they give to draw the diagram, you have to draw this both the things. First you have to draw the outline like this, then you have to show the magnified structure of the layers, okay, of a anther. And also remember, you have to remember the order wise. Okay, that is outermost to innermost or innermost to outermost, because they will be asking in your competitive exam like this, which is the correct option. That is they will give like epidermis to endothelium, your first option, and then your then they will give middle layers. Okay, then they will give tapetum. In other option they will give first endothelium, then they will give middle layers. Like this different question will be asked on this, so you just be clear with this. Okay, the layers. So remember, the middle is a connective tissue, and then we have outermost epidermis, and next to the epidermis we have endothelium, and next to the endothelium we have the middle layers. Layers means you have to show the two layers, and then we have tapetum. So this outermost three layers, that is epidermis, endothelium and middle layers, is used in the case of protection, whereas tapetum is a nutritious layer which is used to give the provide the nutrition for the developing pollen grains in the sporogenous tissue. This is a sporogenous tissue. Okay, you have to label properly, and you have to show this magnified diagram. Okay, and remember, you draw middle layers, two layers. Okay, and remaining epidermis draw one layer, endothelium draw one layer, middle layers you draw two layers, and then you draw the tapetum. And please don't forget to put the dots. This is a nucleus, that is showing that the tapetum is a nutritious layer. I think hope you understand. You don't need to draw this particular diagram. That is, this particular diagram is not required to draw. This is only for your understanding, but these two diagrams is very important. So now let us go for a fourth diagram. So this is a next diagram, that is enlarged view of a pollen grain tetrad. So see what diagram this is representing. This is representing the stages of the microspore developing into the pollen grain. What is microspore? What is pollen grain? See, we know that. See, I just take this diagram. See, in this particular diagram, I told you sporogenous tissue is present. Can you see the sporogenous tissue? This particular circle is a sporogenous tissue, right? So in that particular sporogenous tissue, there are many microspore mother cell. There will be many microspore mother cell. That particular microspore mother cell will undergo meiosis. Can you see? I have drawn the flow chart. So first you have this particular sporogenous tissue. Can you see the sporogenous tissue here? So this is sporogenous tissue, and that sporogenous tissue consisting of many microspore mother cell, and that particular mother cell will undergo meiosis, which will give four functional microspores, and that will get matures into pollen grains. So this is a stages of the maturation. See, what happen is, you have the microspore, right? See, you have this particular microspore, right? This is a microspore. Okay, so inside the microspore we will be having vacuole. Obviously it's a plant cell, obviously it will be having vacuoles, and one nucleus will be there. That particular nucleus will become asymmetrical spindle like structure, because the division should takes place. Now what division will takes place? Mitosis will takes place. Here it will undergo mitosis. Okay, what is that? Mitosis. Remember, mitosis will takes place. Mitosis will takes place. What will happen inside the microspore? It will be divided into two cells. One is vegetative cell, another one is generative cell. Generative cell is a small cell, okay, which will float inside the vegetative cell. Okay, whereas vegetative cell is a bigger cell and a very nutritious cell. Okay, this particular generative cell, okay, it will be next once again undergo mitosis and divides into two male gametes. So pollen grain itself is a not male gamete. Inside the pollen grain, that two generative cell will undergo once again mitosis and will produce as the two male gametes. So hence, to make a male gamete, it should undergo one meiosis, okay, and two mitosis. So where it undergo mitosis? Here one mitosis and here one mitosis. So like this, one meiosis and two mitosis is required for making the male gametes. Next diagram. So in this particular four diagrams, the D diagram is, that is the diagram of the anatroposovial. Very important for your examination purpose. They will ask to draw and as well as labeling. Okay, I will explain you later. Then what is this? They have simply given, this is you already know, it's a gynoecium, that is stigma. You have the stigma where the pollen grains come and attaches there. Then the style, it's a path of the pollen grain where it will travel towards the ovary, and then we have the ovary and the thalamus. Okay, I think hope you understand. Next, they have given the two example, that is the different types of the gynoecium they have given actually. That is in the case of multicarpellary pistil, that is many carpels will be there. In that case, it may be fused or it may be free. If it is a fused, it is called a syncarpus. That particular syncarpus you can see in the papaver plant, that is poppy seed plant, what we call it as guscus. Okay, and next we have this particular flower, that is a Michaelia champaka. Okay, this is a flower. In that particular flower we have the carpel which is free, that is called as apocarpus. Okay, they have given the example of the syncarpus and apocarpus. Okay, syncarpus and this is a apocarpus. Okay, and next your diagram, very important diagram. Anatropous ovule. Why it is called as anatropous ovule? This is because, see, due to the placental elongation, there will be placenta. Okay, due to the elongation of the placenta, so that ovule is there, no, that particular ovarian body, that is ovule body, is little bit curved, okay, by 180 degree. That is why it is called as anatropous. Okay, I think hope you understand about it. Now let us go to the diagram. So first you have this particular funicle, where funicle is the stalk. Okay, that is a stalk which is used to hold that particular ovule body. Okay, where hilum is a fusion of the funicle, that is a stalk, and the complete ovule body. Okay, and next is your micropyle, where a small opening where some oxygen will enters and some transpiration will takes place, water will enters through that particular micropyle. And next we have the micropylar pole. Okay, and we have integuments, that is the outermost covering, that is we have outer integument and inner integument. Okay, and in the case in the middle we have nucellus. See, this is outer integument. Okay, and this is inner integument. In between this particular place is there, no, this thing, this is the nucellus. This is a nucellus. In that nucellar cells, one of the nucellar cell become very special, and that is megaspore mother cell. See, you have the nucellus. In that nucellus many cells will be there. In that one cell become very special. Okay, that become very special and will be converted as megaspore mother cell. That megaspore mother cell will undergo meiosis. After it undergo meiosis, it will obviously form 4 cells. In that particular 4 cells, one cell become a functional megaspore that will be developed into the embryo sac. So can you observe the embryo sac here? Let me show that one. This is the nucellus. This particular part is there, no, this thing, this is a embryo sac. I think hope you understand. You have that particular megaspore, right? Now that particular megaspore should be converted into embryo sac, right? How that particular embryo sac is converted, that is the steps that they have given here. So let us understand the steps. This is what I explained, that in the case you have the nucellar cell, one cell becomes special and that is undergoing meiosis here, and megaspore tetrad, say 4 cells, that only one cell becomes special. Okay, see, I think hope you will understand here. One that is functional megaspore is there, it will undergo mitosis, one will become 2. So next, once again it will undergo mitosis, so 2 will become 4, that is double, right? And 4 is there, another one mitosis will take place, which will become 8 cells. So in this particular 8 cells, what will happen is, so your 3 cells, let me draw your, in that particular 8 cells, so 3 cells, okay, will go one end, another 3 cell will go another end, where 2 cells will remain in the middle. Okay, so hence these 2 cells will combine to form one polar nuclei. Okay, okay, so I think now I draw the bigger one. Yes, can you see these 3 cells which will go towards the chalazal end is called as antipodals. Okay, antipodals. Okay, and the 3 cell which come towards the micropylar end is called as egg apparatus. Okay, in that particular egg apparatus, 2 cells, okay, will be synergids. Okay, synergids, and one cell will be egg cell. Remember, don't be get confused. So egg apparatus is not mentioned in the NCERT textbook, but it is very important to remember. The 3 cell which come towards the micropylar end is called as egg apparatus. In that egg apparatus, 2 will be synergids. Okay, 2 will be synergids, whereas one is called a egg cell, where your particular pollen grain should come and fuse. Okay, the next diagram is very easy, that is the pollen grains which is terminating on the stigma. See, the pollen grains will come and fall on the stigma, which is a stigmatic surface. Okay, very sticky surface. Okay, and what happens is, not every pollen grain will develop its pollen tube. Okay, only the compatible pollen grains is there, no, which is have ability to develop the pollen tube. It will develop its pollen tube. So pollen tube is a long slender, the slender like structure, which consisting of the pollen grains. Okay, so it will develop and enters the ovary. Okay, and enters into the ovule through the micropilar end. Okay, can you see the micropilar end? So can you see this one? So two synergids is there, one axil is there, pollen tube will come where it will be carrying the pollen grain. In each pollen grain we have two male gametes. So one male gamete will go and fuse with the axil, which will form zygote and embryo. And whereas another male gamete will go and fuse with the polar nuclei. So polar nuclei which was there at the center, it will go and fuse with the polar nuclei, which is called as triple fusion to form the endosperm. And this particular endosperm is very essential to provide the nutrition for the developing zygote, where one gamete fused with this egg cell which forms zygote. So for that developing zygote, nutrition is essential. Yes, that is there, which actually showing in the diagram, that is male gametes entering through micropilar end, where the filiform apparatus is helping that, where one, see, can you see, observe this particular diagram. Let me zoom it and show you people. Can you see this particular, where one gamete is there, no, one gamete is going towards the egg cell, where another gamete is going towards the polar nuclei. That is what they are showing in the diagram. I think hope you understand this diagram. Let us move on to the next diagram. So now let us move on to the next picture, that is fertilized embryo sac showing the zygote and primary endosperm nucellus. Can you observe before the post fertilization events? So zygote should be developed, right? That particular thing is shown here. So we know that in the pollen grain we have two male gametes. One male gamete will come and fuse with the central polar nuclei, which will forms the endosperm, whereas another male gamete come and fused with the egg cell, which form the zygote. So the main intention was the formation of zygote that has happened. Now the embryo sac will loses its structure. It means that, so the synergids will get degenerates, antipodal cells get degenerates. Okay, remaining thing is your endosperm as well as your zygote will remains. So what happens the fate of the zygote? So zygote, it from one cell it's go on dividing. Okay, so can you observe? So zygote to proembryo, proembryo to globular embryo to heart-shaped embryo, heart-shaped embryo to mature embryo. That is what they have shown here. So you can see the suspensor cell. So in heart-shaped embryo, you can see the formation of suspensor cell here. Here also we have. So suspensor cells is mainly used to derive the nutrition, okay, from the endosperm towards the zygote. Okay, it will mature embryo. You can clearly see the radicle and cotyledon and plumule. So you can see the plumule, that's the shoot tip, whereas radicle which will move as a root tip, and the cotyledons, that is, you have the cotyledon, you know the cotyledon, right? So that is the diagram. I think hope you understand this. So your primary endosperm is a 3N, that is not a haploid, not a diploid, it's a triploid. Okay, it's a triploid. That is important to remember. They will ask the MCQ like this: primary endosperm will be having which ploidy? So you should know that is a triploid. Okay, now let us go to the next picture, that is they are showing a typical dicot embryo and a longitudinal section of an embryo of grass, that is monocot. Grass will belong to the monocot family. So coming to the dicot, can you observe the dicot here as they have shown? So two cotyledons will be there. Dicot means obviously two cotyledons should be there. Plumule, that will act as a shoot tip. So if I consider a plant, obviously there is a shoot tip, root tip. Shoot tip is the precursor, okay, of the shoot tip is plumule, whereas the precursor of your root tip is radicle. Okay, remember like this: R for radicle, R for root. Okay, and then remaining is your plumule, which will act as a shoot tip. And then you have the root cap, which is a thimble-like structure which will helps to protect the root tip, and hypocotyle. So next diagram is the embryo of the grass, that is you can say this is a monocotyledon family. So in the monocotyledon family we can see only one cotyledon. So can you observe the one cotyledon, that is the cotyledon, that particular cotyledon, and this is very very important, so don't forget this one, that is cotyledon of monocot, okay, of monocot is called as scutellum. You must remember this one. The cotyledon of the monocot is called as scutellum. That is already shown here. Only one cotyledon we have. So, and next we have the coleoptile. See, coleoptile is the covering of the shoot apex. Okay, and then similarly we have the root apex, that is radical. Okay, so for the radical protection we have coleorhiza. How to remember? Radical protected by coleorhiza. So coleorhiza, R, we have a R, remember this particular R. Okay, so coleorhiza. Similarly coleoptile, that is plumule, is protected by coleoptile, P. Okay, so like this, and remember this is a epiblast. So what is this epiblast? See, first which plant came first, the monocot or dicot? You have a doubt in this, right? See, remember the first plant which came was dicot. So on evolution dicots become monocot. So dicots means they were having the two cotyledons, right? So on evolution the one cotyledon got reduced, and it got reduced to what? Epiblast. The reduced cotyledon is called as epiblast. Remember this one. So the reduced cotyledon of a dicot is called as epiblast. We were having the two cotyledons. One, it was called as scutellum. Okay, another one got reduced and turned into monocotyledon, and another one became as a epiblast, which become a non-functional, and it's a remnant of the so that is dicot. It's a remnant of the dicot cotyledon. Understanding this one? So this is very important to understand. That is, one was the scutellum. The one was scutellum, what you have to understand, and another one is epiblast. Remaining everything you know. Okay, I think hope you understand about this diagram. Okay, fine. So now let us move on to the next diagram. So yes, can you observe this? See, here you can see the many diagrams, but they will not ask you to draw all these diagrams. Okay, so the diagrams is not asked to draw, but in the case of competitive exam they can give easily. They can give where they will give the diagram and they will give some missed parts. Okay, where you have to label according to the number. For example, they will give your A, they will give your B, and it will be given in the form of match the following, like what is A, what is B, like that. That is very important in this particular diagram part. So I will explain each and every diagram here. First let us go to the first diagram. Can you observe the first diagram here? So we have, this is very simple. You have a cotyledon, that is obviously zygote develop into embryo, and dicots, if it is there, means two cotyledons will be there, right? So that is there shown dicotyledon. So similarly, if it is in the case of seed, okay, so micropyle will be there, that is a small opening. Okay, small opening will be there. I have already said about the micropyle. Okay, so that was about the first diagram. And second diagram is also very easy. So that is they are giving the different types of seeds, that's it. Okay, so you can see the seed coat. Obviously you know that seed coat will be there for a seed. In that seed coat, you remember the first year concept, that is outer is called as testa and inner layer is called as tegmen. Okay, seed coat also we have two types, testa and tegmen. Okay, remember that one for the competitive purpose. And next you have the cotyledon. Okay, so that is, you know, the cotyledon. And then you have a shoot apical meristem, that is obviously the shoot. Obviously seeds means plants means meristematic tissue, obviously it will be there. So they are showing the premature one, that is in the seed where the shoot apical meristem located. And hypocotyle we have. Then root tip, can you observe the root tip here? So this is a root tip. Okay, the very end point is, you know, that is a root tip here. And next we have the endosperm. that is the nourishment provider. See, this is a monocot, so that's why endosperm is remnant. But in the case of dicot, endosperm will be not there. Okay, so it will be converted as cotyledons, that's the storage body food. Okay, in the case of dicots, cotyledons will be storing the food, but in the case of monocots, we don't see the two cotyledons, right? So at that time we can see endosperm. So endosperm itself will be acting as a nourishment provider. Understanding? So endosperm will be present in the case of monocots, and dicots we cannot see the endosperm. So next thing, that is this particular diagram. Here also they are giving the like diagram where you can see the seed coat, endosperm, and a cotyledon. You can see the cotyledon and hypocotyle axis and shoot apical meristem, root tip. Okay, that particular very sharp point is there, no? Let me show you. So see, that very tip is there, no? That is a shoot. That is a shoot, whereas the very lower point is root. Okay, I think hope you understand this. So now let us move on to the next part, that is your this diagram. See, I think you know this diagram, you have already know in this first year, right? In the first year you have this diagram. Okay, I think in the morphology of the flowering plant, I think so, in that particular lesson you have the pericarp, you have the endosperm, you have the scutellum, and I already said for the plumule this is very important. They always confuse you in the case of competitive. They will give this as a coleorizer, they will give this as coleoptile, but it's a wrong diagram. Okay, so I've already said that, right? That is, your plumule is covered by the coleoptile, P for plumule, P for coleoptile, whereas radicle is covered by the coleorhiza, R for radicle, and coleorhiza R. Okay, remember like that. And then you have scutellum, that is a monocot seed, and endosperm, obviously in monocotyledon seed we can see the endosperm. And next we have pericarp, that's nothing but it is a like seed covering, that's it. Okay, and next, so they have given about the examples of the false fruits, that is apple and strawberry. So false fruits, know that, see, we know that after the formation of zygote and all, so your ovule is there, no? Ovule will turn into seed, whereas ovary will turn into fruit. So ovule will turn into seed, ovary will turn into fruit. But in some fruits, the fruits will be developed other than the part of ovary, that is from the thalamus. That is the example for, so example for that is your apple as well as strawberry. Okay, and also remember this achene, and this is a thalamus. This part is thalamus. Achene is the seed embedded in fruit. Okay, I think hope you understand about the diagram, so, and also explanation what I have given. I think hope you like this video. If you like this video, please like the video and please share to your friends as well as subscribe to my channel. So thank you everyone, have a great day.